Hello everyone. Today I'm here with my new video which is a quiz on Antisense RNA technology. I'm sure you all must be knowing a little about Antisense RNA technology. I have also made one video which deals with the details of Antisense RNA technology. You can go through that video also before starting this quiz. But if you have already seen it or already know about this antisense RNA technology, then let's start the quiz and I'll give you the link of my video. So the first question is antisense RNA is. So that means what is antisense RNA? It is same as messenger RNA, or it is transfer RNA or double-stranded RNA, or it is complementary to specific messenger RNA. So if you know what is antisense RNA, then you must be knowing then that it is complementary to messenger RNA. It can't be messenger RNA. So first is not possible. tRNA again is not possible because that is a it helps in protein synthesis, but then it picks up the amino acid, so it's not possible. Double-stranded RNA, not at all possible because antisense RNA is a single-stranded one. So the right answer is complementary to specific RNA. Antisense RNA is single-stranded, messenger RNA is also single-stranded, and both are complementary to each other. So our right answer is D. Let's see the second question. Which among the following can silence the genes? Autoimmune diseases, no, these are kind of diseases which are related to your immune system, where your immune system is not working properly and it is attacking the healthy cells, so that is not possible. Gene cloning, again, it's a part of genetic engineering, it's not here. It's not possible for gene silencing. Antisense RNA technology, yes. The answer should be antisense RNA technology. And again, the last one, the chromosome walking, that can also not be possible here for gene silencing. So let's see the answer. Answer is the third one, antisense RNA technology. In this quiz, it is very easy, probably the most easy question, because we are here talking about the antisense RNA quiz. So here the answer is antisense RNA technology. Third one, what is the primary function of antisense RNA technology? So in these type of questions, you must read it carefully again, because here I am talking about the primary function. So genome editing is one. See, genome editing may be the ultimate goal, but it's not the primary function of antisense RNA technology. It inhibits the expression of a specific gene. Yes, that should be the answer. Let's see the answer. Yeah, so that is the answer, but the third one is not possible, enhance the protein synthesis, because it is none of the antisense RNAs can enhance the protein synthesis. On contrast, it is going to stop the protein synthesis, and gene mutation has nothing to do with the antisense RNA technology, so that is also not possible. Fourth question, antisense RNA can block gene expression by, that means here we are talking that how the antisense RNA blocks the gene expression. You know that antisense RNA is single-stranded RNA, and then there is messenger RNA. Messenger RNA is the one which forms the protein. So if you have to block the gene expression, or for that you have to block the protein synthesis, then the messenger RNA should be stopped. So how it is done? Forming the stable duplex. So that should be the right answer. And preventing transcription, no, transcription is, you must be knowing, the messenger RNA formation is the transcription, so that is not possible. Blocking amino acid synthesis, no, because amino acids synthesis is a separate topic. And preventing replication of DNA, no, not at all. So all the last three are not possible. Forming stable duplex is the right answer, because here messenger RNA and antisense RNA because they are complementary to each other. So they make a pair, and when they make a pair, they form the duplex. And in duplex, RNA is never in two strands. It's in double-stranded form. RNA is never in double-stranded form. So in this case, protein synthesis is not formed. Fifth one is antisense RNA is different from RNAi technology because, so here I would say that RNAi technology is another very important technology which is a little similar to antisense RNA but definitely not the same. To know the answer, you must know about the RNAi technology. And I have made 3 videos on RNAi technologies also, which are related to siRNA, mRNA, and shRNA. So I'll give the link of those videos also. Here, the first option is, it is siRNA. No, it is not possible because it is the part of RNAi. It is complementary to single-stranded RNA. Then it is synthetic double-stranded RNA. Double-stranded, that is not possible. Yeah, it is short hairpin. No, that is also again not possible. So let's see the answer. Answer is the B. It's very easy for you to answer this question if you know what is antisense RNA. So it is complementary to single-stranded RNA. Most of the time we are saying that it is complementary to messenger RNA, but here we are saying it is complementary single-stranded RNA. So it's almost one and the same thing. The answer is the B. Sixth one, which among the following is true about antisense RNA? It has RISC complex, it has dicer, it is highly specific as RNAi, or RNase H is there. So, let's see. RISC complex is a multi-protein complex which is there in RNAi. So if you know little about the RNAi, it will be very easy for you to know that RISC complex is present in RNAi. So that cannot be the answer. The second one is dicer. Dicer again is an enzyme which can cut double-stranded RNA. So here in antisense RNA technology, double-stranded RNA is not there. Then it is highly specific as RNAi. Then antisense RNA is not as specific as RNAi. So RNAi is a better technique. So that can also not be possible. And RNase H, this should be the answer. So RNase H, this can, single-stranded RNA can be degraded by this. So this should be the answer. And here, that is our target. The target is messenger RNA. Messenger RNA is single-stranded. So that we have to cut or degrade. So RNase H can do that. Now let's see the next question. What happens when both sense and antisense RNA are present in the same cell? So here we are saying that antisense RNA and the sense RNA both are present in the same cell. Now, do you know what is sense RNA? If we are talking this RNA as the antisense RNA, then there must be some sense RNA also. Here the sense RNA is messenger RNA. And why messenger RNA is called as sense RNA? Because messenger RNA is the one which makes the protein. That is the main function of the gene. Gene makes the protein. That's how it, controls the whole thing. So that's why messenger RNA is known as the sense strand or sense RNA. So let's see the options. It causes mutation? No, mutation is something different. Initiate translation? No, because antisense RNA technology, again and again, we are saying that this is for stopping the translation, not to initiate. Then third one is form a RNA-RNA duplex. Yeah, that can be the answer. Let's see. Yeah, this is the right answer because here One RNA is antisense RNA and the second RNA is the messenger RNA. Both are single stranded. Since both are single stranded and both are in the same cell, so they will come close and they will form a pair, and they'll form a duplex. So this is the right answer. And the last one was become carcinogenic. No, carcinogenic is cancer causing. No, that is not the right answer. So the right answer is C. Which organisms have naturally occurring antisense RNA? So here I actually wanted to tell you that antisense RNA are not only man-made, not only they can be synthetically prepared, but these are naturally present also. So here the options are humans, bacteria, archaea, and all of the above. So the here answer is all of the above. And not only human, bacteria, and archaea have naturally occurring antisense RNA, but there are plants, animals. I mean, all kind of organisms have these antisense RNA which are naturally present. So they have their function when they are naturally present. And in this question, there is another thing that all of the above is one of the option, and most of the surveys says that when this all of the above is one of the option, the chances are from 25 to 50% that this one can be the right answer. But of course, before guessing or just taking the risk, you should be very calculative. So anyways, the answer is D. The next one is, which one is not true about naturally occurring antisense RNAs in the cell? In this question, also you have to read it very carefully, otherwise you will not be able to answer it, or you may be confused. So here I am talking not true. So let's see the options. It can enhance the protein synthesis. Can it? Can it enhance antisense RNA? Because antisense RNA, binds with the messenger RNA and interfere with its function. So it cannot actually. And second one is it can bind to target messenger RNA. Yes, it always binds to messenger RNA. So that is true actually. It can regulate the gene. Yes, it regulates the gene. It can inactivate the X chromosome. Yes, that is also done. That is also one of the function of naturally occurring antisense RNA which is present in the cell. So that means here B, C and D, these are all true. But the question is which is not true. So the answer should be first. Next question is, for gene silencing by antisense RNA, which of the following is given from outside? Now here we are not talking about the naturally occurring antisense RNA. Here we are talking about the technology when for gene silencing we are providing the antisense RNA from outside. That means if we are providing it from the outside, we have to prepare the antisense RNA in lab. So that's why I have written oligonucleotides. So these are all synthetically prepared oligonucleotides which in the form of antisense RNA will provide to the cell. Now let's see the options. Only sense oligonucleotide. Sense, I just told you that sense RNA or sense oligonucleotides are the ones which we call as the messenger RNA. And only second one is only antisense. So antisense oligonucleotides means the antisense RNA which has been prepared outside in the lab. So that should be the answer. And then the third one was both sense and antisense. This option has been given to you just to confuse you that maybe both the strands are needed, but no, both the strands are never needed in antisense RNA technology. In RNAi technology, we use double-stranded RNA, but never in antisense RNA technology. And none of the above is the fourth option, which is here it is not true, and otherwise also, I mean, in most of the cases, none of the above is not the right option most of the time. But of course, you can't be very sure. Antisense RNA was used in producing. The options are golden rice, BT cotton, flavor saver tomato, and virus resistant papaya. So here we are talking about the applications of antisense RNA technology. Golden rice, BT cotton, I mean, all of these are very important products of genetic engineering. Golden rice, you must have heard about. It's yellow in color and it is rich in vitamin A. So that's why it is yellow. So no, that can't be the answer. BT cotton, again, BT cotton is also a transgenic plant in which the bacterial bacillus gene was incorporated. But no, here it can't be the option. Flavor saver tomato should be the answer. And virus resistant papaya again is a disease resistant papaya crop, which is a transgenic crop, but it has never been produced by antisense RNA. So the right answer is flavor saver tomato, and probably this one is the most important application of the antisense RNA, most highlighted one. Now the last question, which is the target enzyme for producing flavor saver tomatoes? So this is about the target enzyme of the flavor saver tomatoes. 4 enzymes are given in the option: polygalacturonase, amylase, cellulase, and lipoxygenase. All of them are very important enzymes in their own way. But here I must tell you that mostly, I mean, there are many industries which are based on tomatoes. And with tomatoes, one problem is there that they are softened very easily and in a few days only. So they have very little shelf life. So because of that, it was thought that if we have to make the tomatoes for the industry, then they should survive for a long time. They should not soften. And for softening, there is one enzyme which is polygalacturonase. So let's see the answer. Yeah. So this polygalacturonase is the enzyme which is the culprit, or which is the one which makes the tomato soften very easily. So if this enzyme is not present there, or it is reduced in amount, then these type of tomatoes can be very useful for the industry. So keeping that in mind, these flavor saver tomatoes were formed, and here the target enzyme was polygalacturonase. So that is the right answer. So the messenger RNA which formed the polygalacturonase enzyme, that was the target RNA. And if they ask you the gene, ask you the enzyme, then the polygalacturonase is the right answer. So that is all about this quiz right now. I will again tell you this thing that it is very important to go through the videos or the chapter about the antisense RNA technology. Very important technology it is, and it has many applications. So you can go through my video, which the link is given, or otherwise any other video or any other book where you can find this chapter. So that will help you to understand all the questions. And if this information was useful for you, then please subscribe my channel so that you can see more quizzes. I have made many quizzes on different topics, so that will be very useful for you. Thank you.